panel on ancient DNA. And I want to first of all thank the panelists for agreeing to be here. And I'm going to run through a brief introduction of each of them, and then we'll have each one come up and give some remarks and then open up for discussion. But I also want to thank all of you for being here and participating in our discussion. And hopefully this is something that we will continue to think about as it evolves a technique in many different areas of social and natural sciences. So we wanted to highlight that. So first of all, our speaker from earlier today, we built this panel around, Sabina I can't do it. Cveček, too late in the day, is the Marie Skłodowska-Curie Global Fellow at the Field Museum in Chicago and the Austrian Academy of Sciences in Vienna. Her research explores prehistoric kinship, household organization, and social practices of relatedness beyond genetics, integrating archaeological, bioanthropological, and ethnographic perspectives. She holds a PhD in sociocultural anthropology from the University of Vienna. She has been a visiting researcher at the Max Planck Institute for Social Anthropology and an IFK Junior Fellow at the International Research Center for Cultural Studies in Vienna. She's an elected fellow of the Royal Anthropological Institute and a co-chair of the Archaeology and Gender in Europe network of the European Association of Archaeologists. Her recent publications include Why Kinship Still Needs Anthropologists in the 21st Century and Enthroned, Dethroned, and Rethroned ? Multiple Lives of Matrilineal Kinship in Aegean Prehistory. Second, we have Manjusha Chintalapati. Oh, I don't think so, but thank you. Works in bioinformatics research, currently doing postdoctoral work at Ancestry DNA. She has an informatics PhD from the Max Planck Institute for Evolutionary Anthropology. In 2019, she joined the Morjani lab at UC Berkeley, where she continued research on human population genetics, developing methods to infer demographic events such as timing of admixture and workflows to infer germline mutation rates. Her current research focuses on analyzing human populations, including present-day and ancient groups, and developing computational methods to better understand human diversity. In addition to basic research, her scientific interests include translational projects, including leveraging genomic data for phenotype and disease mapping. Beyond research, she's passionate about storytelling and science. She loves turning data into art and even paints her own scientific posters. Very impressive. Next, at the end of the table here, we have Sophie Joseph, who's a postdoc in the Morjani lab in the Department of Molecular and Cell Biology here at UC Berkeley. She specializes in studying human evolutionary history around the world using both ancient and modern genomes, reconstructing evolutionary events like natural selection and ancient admixture between human populations, and even between different hominin species. She received her PhD in biological anthropology from Emory University in May 2025, newly minted, congratulations, working in the Lindo Ancient DNA Lab. And last by no means least, Gypsy Price is a biological archaeologist who currently serves as a lead principal investigator for Search LLC, a cultural research management firm operating worldwide. In addition to planning and managing excavations throughout the continental United States and the Caribbean, she also moonlights as the lead biological archaeologist for the Nemea Center for Classical Archaeology excavations at Petz's house, Nemea and Ivonia. Maybe a reason why I tapped her to come and talk about. And she, I will add, has been also responsible for doing our aDNA sampling and working with the Max Planck Institute on those results of ancient DNA from our sites. So, I don't know, Meg, you're just gonna moderate, just the time person. I don't know if we have do we want to go in?
I think the slides, each one of them.
You guys have did you put them together in a way that reveals to us how you're gonna present?
So we'll start with Sabina.
Okay, perfect.
So we'll go there.
Perfect.
Sabina,
I didn't consult the order.
Sophie and then Gypsy.
Okay, perfect.
Order, and
Perfect.
So
Thank you. So we'll start off with Sabina. Welcome.
If I can move this. Oh, okay. Okay. Good evening, everyone, and thank you so much for having me as part of this panel. I see some familiar faces, so I didn't scare you away earlier in the day. I'll be talking shortly in this 3 minutes presentation about interpreting ancient DNA from anthropological perspectives, as I'm a trained cultural anthropologist who worked since 2016 with archaeologists, and now I'm starting the third year of my AXKIN project, which looks at prehistoric kinship from sociocultural anthropological perspectives, integrating ancient DNA data, but also other things. So as one of the examples that I would like to bring in here is kind of this example from Gurgy in France, a 7th-generation pedigree that was reconstructed from a Neolithic cemetery, which marks not only the 2 large genetic pedigree families, but also a lot of unlinked, unrelated individuals. And this is what I am after in my interpretations of prehistoric kinship, to kind of highlight what can we do with those things that don't really fit necessarily to the kinship or genetic pedigree that is necessarily constructed by geneticists. In my AXKIN project, I look at 2 questions. First one, how can material structures such as settlements, figurines, or buildings, artifacts, and biological markers be read as material codes of prehistoric kinship? And second, how can ethnographic reports exemplify rather than verify variability in kinship during prehistory? And some of those questions I outline in the paper of why kinship still needs anthropologists in the 21st century, published in Anthropology Today. I looked at the 4 Neolithic and early Bronze Age sites in southeastern Europe and Anatolia, namely Vucedol, Lebenskivir, Çatalhöyük, and Aslantepe. And all of these sites have intramural burials, the so-called residential burial within houses, where then it's not just the cemetery as a unit of analysis, but really the house and how those burials are related within those units. And just to highlight one example, which I have brought to the talk today already, there is a case from Çatalhöyük of 4 infants buried next to each other, but they are really not genetically related. And reasons for that can be many. And this is something that we unpicked very shortly in a recent publication, Beyond Genetics, exploring aspects of non-biological kinship in prehistoric times, based on a 2-day workshop in Vienna, in which we really invited archaeologists, cultural anthropologists, and geneticists to think about what other ways than genetics or biological links can be there to reconstruct making kin in the past, and to really bridge new links between archaeology, archaeogenetics, and cultural anthropology when thinking about kinship. Thank you very much for having me here, and that's all from me for now.
Main theme is, just getting up here to do a little bit, the main theme is advertised is that we're really interested in how people interpret what the results they get. We're not talking today about methods per se, although methods may be part of getting to an interpretation. But what is it that different researchers can bring to the challenge of interpreting the results that we're now getting so many of in so many ways in so many places? So, thank you so much.
Yeah. First, thanks to the organizers for having me here. I know I'm working on a completely different aspect of the whole reconstructing history. I'm a geneticist. My work starts after the hard and tedious work of everybody sitting here. I know that archaeologists do the fantastic job of getting the samples and getting DNA, and I work with the genetic data. So thanks to you all for getting my research, and I spent the past decade here. I did my PhD in Max Planck, where I did genomic analysis. So most of my work was actually working on comparing humans with Neanderthals. I worked partially also on Denisovan genome, but most of my work has been on the Neanderthal genome. And when I started PhD back then, we only had the high coverage Altai genome, and now we have 3. I've been luckily part of 2 of the high coverage consortiums, so it was fantastic to see how consortiums work, how much work goes into it, the tons of groups working to get the genome together and make the results robust. I'll not bore into details like Mick said. So very briefly, because all of you are coming from archaeology background, I want to say what I actually work with. I work with DNA segments themselves, like the DNA strands themselves, or the ATGCs. But the interesting thing is what we do is we compare human genome with archaic genome, and the DNA is degraded, as you all know already, better than me. What we do is we are looking at these small segments, small sites which are different between them, and also sites which are missing, which we call indels. That's where the whole comparative genomics comes in. It's interesting to find the sites which are different, which are called SNPs, and these are called indels. They carry a lot of biological meaning, because that means that because we have been separated for so long, each genomes have evolved on their own, and each of these sites can actually play a role in phenotypes like differences in skin or differences in immune, things like that. So a very simple reconstruction of the tree. I, in my PhD, looked at sites or the SNPs or indels, which are only found in humans, and sites which are shared between us, and then which are introgressed, and then look for some of the phenotypes. But in Priya's lab, what I did specifically was looking, using genome chromosomes itself as a marker. So all of us have, let's say, when you have like admixture happening between modern humans and Neanderthals, which we know for a fact now already, the first generation inherits one chromosome from each, and then over time passes, they break down because of recombination. We already know that. But recombination is a beautiful natural thing, as we already also know that. But as a geneticist, it's very interesting for me because it's like a clock. It's a very beautiful clock, because just by measuring the segment length, we can tell how far back the admixture happened. So based on the length, if it's a short one, that means it's been long, and if it's a long block, it's younger. So just using this basic intuition, we developed two methods in the lab. I'm not going to talk about all of them. Talk briefly about one of the recent work that we have published last year, where we applied this on approximately 300 genomes, and all the way from 2,000 years to 40,000 years. And this is just a representation. And a 40,000-year-old, you will see the segments are much longer, as I said, because the recombination didn't have much time to break it down yet. In a present day, they will be shorter. And we looked at several aspects. Again, there's much to talk about it, but very, very briefly, what we looked at is the time of admixture, how much sharing there is between humans and archaics, and most importantly, what did they do for us? Like, what kind of genes did they give us? Was it advantageous? Did we benefit from them? We did. We have some immune genes that we got adapted to that were beneficial for us. And there were some genes which were lost over the time because probably they were not useful for us. And we also, using combined data, we could actually also narrow down the time of admixture and some selected analysis. But to all this, the amazing and best part of it was when we were doing this paper, we had this time, we had this beautiful data set. And at the same time, Max Planck had another paper from Johannes' group, who published high coverage genomes from Germany and Czech, that is Ranis and Zatkun, and they were very close to the sampling time. So basically, if the admixture occurred 45,000 years ago, they found a sample almost close to that found the same finding, which aligned with the result that we had. For me, it was like, I don't know, in India, we say that there are multiple paths to the same God. It was the same truth. We are all finding the same thing, but we use different paths. And as a geneticist, as a computational biologist, we build these tools which work with degraded data, we test on simulations. And then there is another part, which is archaeology. And using tools, probably they use the same tools, but it's beautiful to find evidence. So I see archaeology and genetics as two eyes. You can definitely see the picture with one eye, but with two eyes, the picture is much clearer. So I think they both go hand in hand. And even in my older paper dates, we always use archaeology to understand what the genetics actually gives us, because it gives a perspective to the results we have. And I think they always go hand in hand. And I really appreciate, that's why when you said you were making these bridges, it's so important, and it's so important to get a finer picture of our history. So that's what I wanted to say. Thank you.
All right. Hi, everyone. Again, I'm Sophie. Like Manju once was, I am a postdoc in the Morjani lab, and I also am currently studying archaic ancestry in human populations. But I actually did a lot of my PhD work in the Andes of South America on much more recent human evolution, so within the past 10,000 years or so. And so what I'm going to talk about today is kind of in the context of that conceptual shift that I've had to make in the past 4 months since I've started here at Berkeley. Because as some of you may know, the Andean archaeological record is pretty spectacular and rich. And I could not have done any of my PhD research without my archaeological collaborators providing context into how major demographic shifts in the region are really reflected in our genetic research. But what's been interesting for me to realize since I've gotten here to Berkeley is that when you start comparing different species to each other, like humans and Neanderthals, for example, and the archaeological record gets more sparse because Neanderthals have been extinct since 40,000 years ago, that we can't rely on our archaeologists as much to help us. And so interpretation becomes more difficult. So I'm looking forward to unpacking those sorts of themes with everyone today. So I thought I would just give 3 or 4 slides on some of my current work, and then just kind of make the points I want to make about it. So as far as we can tell, the migration out of Africa that contributed to all modern day non-African humans on Earth happened around 60,000 years ago. And shortly after that, as people wandered into the territory of Neanderthals, we admixed with them about 50,000 years ago. Every single non-African human alive today shares evidence of this admixture event within our genomes, which is quite cool. Yet, puzzlingly, the patterns of Neanderthal ancestry that are held across human populations today are very diverse, despite all stemming from this one admixture event, which is quite puzzling. So this map at the bottom, I hope you can see, okay, is showing in different colors different levels of Neanderthal ancestry that are actually found across modern day human populations. So these darker reds that you see kind of in East Asia and in Oceania are upwards of 2% Neanderthal ancestry in genomes, whereas as you get into West Eurasia, it's more like 1.5%, which seems kind of small, but for context, that's many, many megabases or millions of base pairs of DNA. It's quite a substantial difference. And then there's a very tiny amount in Africa, mostly from backflow from Eurasian genomes. But as we know, the admixture event happened outside of Africa, so that's why Africans have so little. But anyway, so these patterns are really puzzling across Eurasia. And so that's a big part of my research, is investigating specifically in East Asian populations, is my particular work, why some of these patterns exist. And so as someone who worked a lot with ancient genomes in my PhD, my first thought was, well, why don't we ask these ancient genomes how much Neanderthal ancestry they had? So I've actually been working since I've gotten here to Berkeley to apply some of our archaic ancestry inference methods that were used, like for instance, to create this map in modern people, and just applying it to ancient genomes instead to assess how much Neanderthal they have in them. And what we found is kind of an interesting pattern that I think relates to some of the really deep ancestry that we find across Eurasia. And I'll explain to you what these terms mean in a second. But basically, as we were leaving Africa very early on in that lineage, like just after 60,000 years ago, this little group split off called the Basal Eurasians, and they missed out on the Neanderthal fun that was had about 50,000 years ago. But as it turns out, these Basal Eurasians re-admixed into Western Eurasians later on. So not only do Europeans and West Eurasians have Neanderthal ancestry, but they also have ancestry a significant amount from these Basal Eurasian Homo sapiens as well. And so we can see the footprints of this also in ancient genomes, which is what I'm showing you here. So on the left, we're looking on the y-axis at the proportion of Neanderthal ancestry in the genomes. And then these are just a few different ancient samples that are colored by their population. And what I want you to notice is that these guys on the left have like more like 1.5% Neanderthal, whereas over on the right, these two pink and blue groups have over 2%. And there's a significant aspect that's very important to realize. It's the amount of Basal Eurasian ancestry that we think is probably driving this difference. So we'll talk about the interpretation piece of that in a second. So these guys on the left, these early European farmers and these Caucasian hunter-gatherers, they have as much as 50% of this basal Eurasian ancestry who missed out on the Neanderthal lineage completely. Whereas these guys on the right, the European hunter-gatherers and East Asians, they have no basal Eurasian ancestry as far as we can tell. So it seems kind of obvious that maybe that basal Eurasian ancestry who missed the Neanderthal admixture event is like diluting this signal in these Western Europeans. But this is only 10 genomes. So this brings me to my first interpretation point, which is that this could be a coincidence because genetics is very complex. Population movements are very complex. It's quite possible that there's other processes like genetic drift or selection or something else that's actually driving these patterns, not basal Eurasian ancestry. And so the issue with interpretation is how do we prove this connection between this basal Eurasian ghost lineage that we don't even have an ancient genome for and the patterns that we see in living people. So that's something that we're kind of wrestling with. And when you don't have lines of evidence from archaeology, because nobody knows what basal Eurasians looked like 25,000 years ago or what their material culture was, how can we begin to feel confident in making assertions from ancient DNA? And so often our answer to this is just more genomes. We need more genomes. So instead of 10 individuals, I might try to find 50 ancient genomes from each of these different colors to try to be more confident in my assessment. But ultimately, I think having multiple modes of evidence is best and most important. We can talk about that on the panel about what some of those evidences look like. Also, our genomes contain all of our ancestors, which is really cool. So a human being is not just a data point of their genome, but they also contain evidence of 50,000-year-old admixture events and other cool things like that. So we can kind of use that to our advantage when we're interpreting ancient DNA. That's one of my favorite things about being a geneticist, in fact. And then my third point is that we need each other, geneticists and archaeologists. Yeah, like I said, working in the Andes, I've really, really relied on archaeologists to kind of interpret and make sense of what genetics is telling us, because like Sabina spent so much time telling us today, the material culture record is really the context for what the genome is and how people lived, because your genetics don't define who you are, how you behave, right? So we need a lot of information. So when we're working with archaic ancestry, it becomes tricky because a lot of times we may have some stone tools from Neanderthals at very select few sites, but that's not the same as like knowing what kind of household they lived in and, where they buried their infants, right? Like it's a totally different level of information that's kind of fun and interesting to grapple with. So yeah, I have a lot of perspectives. I worked on recent human evolution and now archaic ancestry, so I'm happy to talk about more of this in the panel.
Hello, everyone. My name is Gypsy Price, and I am not an archaeogeneticist, but I have worked a lot with it over the last 20 years throughout my academic and my current career in cultural resource management. So I have some opinions about what it can do for us, what it can't do for us, and what some of those difficulties can be. I am a bioarchaeologist that specialized in stable isotopes. So I was a chemist with my white lab coat working in clean labs. It just wasn't working with genetic material. I was sampling it for other people to do. But I have worked in the field extensively. And like I said, currently I'm in cultural resource management, which means that I'm doing archaeology, but in the private sector and at a faster timeline, basically. Prior to these last maybe 5 years, my interest in ADNA was really wrapped up in how it can help prove cohesion or like improve cohesion amongst modern populations by showing how we're all related with this research on early hominin species and how there's been lots of sexy times between all sorts of things we used to think were very separate. And then therefore, if someone had more or less of that gene, that meant something. And so I really loved teaching human evolution courses or human biological variation courses where I really underlined the fact that with genetic research, we can really talk to the way that we're all connected in ways that we can't even fathom, not just with us, but also with primates and et cetera. So I felt like ADNA was very important to convince people that we should be nice to each other, basically.
I also was interested in the ways that, since one of the prompts and things that we were going to talk about today, how you can use aDNA not just on humans, but on human tangential things. So things like livestock that can teach us about domestication strategies, the transmission of different types of technologies, and things like lice. I always loved teaching about lice and how we can learn about when we lost some body hair and lice diverged different parts of our body, and how we developed clothing, not just in humans, but in what we consider non-Homo sapien hominins as well. Nowadays, I do use aDNA in my cultural resource management job. Most of it's being done on material from the Caribbean, where a lot of times what we use aDNA for in our phase 3 excavations, which are, like the large scale excavations we think of, does a lot to speak to things like the pots versus people debate, whether or not pots are moving or people are moving, and obviously the answer is both. But it's nice to have aDNA to show us that there is interconnectedness amongst, in the Caribbean sense, there's interconnectedness amongst the islands, where you find the equivalent of first degree cousins, several islands apart across generations. So obviously, people didn't just live on an island. As we know in the Aegean, people are moving across water all the time. So we use that a lot. We also use it to provide connections to local populations in the Caribbean and the areas we're working, to indigenous Taino populations like in Puerto Rico, which helps to drive legislation which protects cultural heritage. So that's kind of how I use it in my current paying position, as it were. But why I'm here, as Dr. Shelton had mentioned a second ago, is because I'm also the lead bioarchaeologist for Topos, dealing with the late Bronze Age chamber tomb cemetery in the northeastern Peloponnese in Greece. And we are involved in this larger study where, with the Max Planck, actually in China, where they are looking at populations from across the Aegean and looking at population movement through time. And it's an ongoing study. There has been a single round of publications that have come out so far, and we are currently waiting for results on additional studies that are happening. For our particular site at Idonia, we currently have results from 3 of the chamber tombs that we have excavated in our last 10 years of excavation, maybe 15 now, I can't remember. Time passes very weirdly as I get older. And then we have an additional 29 individuals that we're waiting for. I thought, since you had given your amazing talk earlier and talking about kinship and gender and how that relates to aDNA, I thought I would give a quick little maybe case study of one of the tombs that we saw that had some fun results, in case it can spur any type of conversation or some thoughts on it. Basically, Tomb 103, which is located way down here, kind of at the bottom of the hillside, is the second largest tomb that we have excavated there. We got reliable results, I should say, because some of this preservation is pretty bad, and all 3 of you who are actually looking at the powder and going, what am I supposed to do with this? We got reliable results from 3 different individuals from 2 different locations in the tomb, and all of which were female, which will come into play in just a moment. There's a large central cyst right in the middle of the back side of the chamber, in which we had primary burials all the way at the bottom, secondary burials piled up on top and shoved to the sides, and then all sorts of stuff. And then we can talk about the stuff if you want to. But at the bottom, one of the 3 primary burials that were right at the bottom, there's a female that we got reliable results from. We also got reliable results from a biological female, who was pushed to the side and who was dated a little bit earlier, who's pushed aside, probably to make room for the 3 that they laid out in the middle. And we also got reliable results from an individual that was interred in a secondary deposit in the Dromos. And this was just a giant jumble of at least 3 humans, likely female, and just so happens that the material or the reliable information, aDNA results we got happened to be a female. So I was like, yes, good job, Gypsy. You know what you're looking at when you're looking at your humans. But long story short, what was fun about these 3 individuals is that this particular individual that was laid out all nicely, and that dates to Labelatic 2A, which is similar to the one in the Dromos, and prior to the one in the top, likely. We can talk about, Kim can talk about the actual materials. But this individual was likely the daughter of the individual that was in the top. And this individual was likely a first cousin to this, and this individual was likely a niece to the one at the top. So putting together our, I mean, obviously ADNA results can only give you first, second, third degrees of relation, but integrating our knowledge of the actual material that was interred with those individuals and being able to date them and provide a chronology and interpretation of them, we can kind of build this little family unit, which is not a household, but it is a cemetery, it's a dead household. And then, of course, you know, the feminist in you is like, yeah, they're matrilineal, yeah, you stayed with your mom and whatnot. But of course, we don't have DNA from the humans. And so there's this whole long conversation that we can have about, you know, is this gentleman a brother? Is he married into this unit, but then has to be interred with his in-laws? Or is he someone who is a cousin? You know, we don't actually know the answer to that, but we do know that there is a strong genetic proximity amongst the females that we have evidence, that we have data for within this tomb. And it does kind of go hand in hand. I was going to point out, especially since you were talking about gendering and the types of, earlier, the types of things that biological males and females are maybe expected to do in certain types of situations. Not only do we have strong evidence of female cohesion in the burial record in this tomb and in several of the other tombs, but we also have fun. We have a fun artifact that is one of my favorites about a female that is doing what would typically maybe be considered a male thing. And I'm just going to, for the sake of time, I'm going to go through these slides and pop out to this little guy. So of course, this is Mycenaean, it's late Bronze Age. Tons of figurines, not my specialty. If you want to ask something about figurines, this one knows what to do with that. But we have this lovely little Mycenaean ox who is being driven with reins by an individual in the back. But typically when you see this in the Mycenaean figurine record, you get the little pinched nose and little flat heads of the dudes, right, that are doing this. And instead you got this little woman with her little close hat, right? That so it looks like a female that's actually driving the oxen and doing the work of a man. So anyway, it's just an example of an artifact that speaks to an integration or maybe a mix-up or of what we would assume men and women were doing in these populations, alongside of the genetic data that shows us that the females are at least related in this area. And I'm going to stop there with the pretty pictures for the sake of discussion. The image.
Well, we're going to open the floor to questions from anybody, but I just wanted to make a comment on what we've heard here with these 4 wonderful presentations. In terms of an array of kind of methods and approaches and sources for interpretation, from the anthropological, ethnographic aspect that Sabina has drawn our attention to a great deal today, and to remember to keep culture in the picture as much as we can. We also have, of course, computational methods, which Manjusha really sort of shows amazing things, and linked also to the kind of pattern recognition, which I think is what Sophie has emphasized here, reminding us of sample size and reminding us of multiple lines of evidence is really sort of crucial as a way in which of working. And of course, again, several of them mentioning how important collaboration is, and looking at, you know, the context, the detailed context within which some of these analyses are working on materials from. And of course, I thought it was great to think, Gypsy, about how even in cultural resource management, ancient DNA information can be really used, and your case study. So we really have basically multi-scalar approaches we've seen this evening, from the macro, multi-generation, large spans of time, all the way down to an individual burial tomb, which shows exactly how wide-ranging and how exciting and so many different approaches that can be used. So that's just a few comments grabbed from 4 very interesting presentations, and we're happy to take questions and comments from the floor. Anybody? Chris, what do you say? We've seen a lot of Bronze Age stuff today. You're a Bronze Age. Yeah.
Well, what's striking to me, Meg, you talk about scale and the scale of analysis, and I owe this to my mentor, Ruth Tringham. The scale of analysis is so important in the different kinds of studies that you talked about. And in particular, I'm trying to wrap my head around this, and you've probably thought about this more. I'd be interested in how scale and things like outliers relate to each other, because in all of these studies, you see things like outliers, and sometimes it's tempting, like I think as archaeologists, anthropologists, we like the dominant pattern, right? We're looking for the pattern, but the outliers matter, sometimes matter even more, or they lead to different kinds of questions. So could you talk about outliers and how that relates to the scale of the analysis that you're doing?
I can make a comment to jump off of, and then I feel like outliers is kind of has been one of your main things. I feel like, okay, so archaeogenetics, we've been doing this since what, the 80s, 84 or whatever, the seminal studies. So again, I'm coming from isotopes, which also began like in the 90s, like in earnest in the 90s, right? And at the time you had nothing, right? You did your 10 samples you could afford. And then you were like, okay, here they are. And then you had to wait for someone else to do 10 samples and someone else to do 10 samples. And then all of a sudden you could actually have comparable data. Here we are 40, what year is it? 25, 40 years-ish later. We're only just now having a significant sample size that we can actually say something statistically that a scientist might actually believe. But I feel like with ADNA studies is that as the years go by, we're going to have less and less of these outliers because we're going to have more and more comparable data. So it's hard to me at this point when we get, so like, for example, if our sample size for us at Idonia right now is 15 or something, and we have 4 or 5 individuals that are unrelated, but like we have hundreds of people there. So are they actually unrelated, or is it just because we don't have the comparable data from those areas? At a larger site, like you're working at where you have many more data points, this might be less of an issue, perhaps.
Okay. Yeah, I'm happy to take the question and take away from you that question of outliers. I think that it's maybe on the one hand scale, but I think that our sites necessarily are not completely comparable, right? Because you have a funerary context versus a domestic context. And so for me, even though geneticists love to compare between sites and like, because these are their few data points to anchor their interpretations, I still think that there is some important difference between, so not that much of it in terms of scale, but the context in which these individuals were found, right? But regarding the scale, what you have just said now, maybe we will see less outliers in the future. But what I think as well, if what we have heard, we want more data and more data points, what we might see is also more regional connections between sites, right? So on the one hand, those people will be still unrelated, but they might have connections that are found, I don't know, 30 kilometers away or maybe 300 kilometers away. And then kind of to show that there was this regional interaction, be that between islands or between funerary placements or whatever. But I think that it's also what you ask in general regarding the outliers. I like to think about Mary Helms or so, in which kind of she also showed that in the Pacific, there were always there would be, or the Marshall Islands, for example, the stranger king. So these outliers, people from abroad or from elsewhere were really parts of communities in different ways, and they could have different social status. They wouldn't have to be necessarily migrants, refugees, the other who doesn't belong, but really people of power as well, and could rule or could have special meaning in the community. So that's my take on like thinking about outliers as a cultural anthropologist, not seeing like the other stranger indeed and like just cut out of all the social ties, but the outlier who can also bring a lot with them that might not be locally cultivated in the community, but they have other types of knowledge or skills or be that social capital or so that they bring with them. Yeah.
I wanted to come back to something that he was talking about earlier today, that which is about enriching the data from DNA with data from isotope analysis. And I'm wondering, none of you mentioned that tonight, but I'm wondering if these ideas of enriching the data from DNA is something that one could do in other ways, beyond even isotope analysis, so that the data that you get, which is presumably from, we're talking mostly about skeletal data, that there are many other things that can be done each time you're taking DNA analysis, which includes how did the person die, how did they get to be now in the place where they are, about the whole cemetery, like of cemetery rituals. I don't know, I'm just wondering what you all think about that in your different ways. Again, I think it probably relates to what Chris just talked about or hinted at with his scale thing.
Okay. Well, I will take just one short example from Archaeology and say that archaeology and just material culture can be really crucial. And that was the case from, I don't know if you're familiar with Archaeology during the late Chalcolithic period. There is a temple and there is the whole storage area and like this grandiose architecture for the time being, kind of the early states of Mesopotamia. And then with the early Bronze Age, the settlement breaks down and it's inhabited by really small houses like wattle and daub and really kind of a pastoral communities or something like that. And the basic question that researchers always were puzzled, Marcella Frangipane and others, was kind of the idea that these must be different population who came in and replaced this late Chalcolithic people. Because you cannot have just this discontinuation and just continue kind of as the same people and reinvent yourself. And there is a paper also by Max Planck Institute in Leipzig, who then sampled both late Chalcolithic and the early Bronze Age material. And these are genetically the same people. And it was kind of a disappointing yet exciting thing to see, oh, right, like you are not genetically reinvented and you're the same group who, because of certain other means of whatever happened in the environment or sociopolitically, they were a dramatic change, and then people started living in a very different way. And this is, I think, a wonderful example of where just like going back to the basics. And I think it kind of highlights the research question, because sometimes, and I know that in genetics, a lot of times the material is sampled without a very clear, maybe research question. But in this case, was really clear. Do we want to see different people or are we seeing the same? And I think this is a beauty where then you can pin it down to just basic archaeological context. And you can see just against genetics, it doesn't live to your own classic expectations.
That's great. I love that example. It reminds me of Maria Gimbutas and the time they are coming. No more beautiful houses, just burials. It's got to be some other different population.
Right.
And then the big DNA people come in and make the big migration, the ones that come from the east of Europe into the west and so on, bring all of our modern populations. And if you look in the details, it's not that at all.
Well, in the reality, DNA is a proxy, right? And you want to do multi-proxy. You're not just going to do everything on genetics.
Yeah.
You're going to also look at
Right.
material culture and the stable isotopes and the yada.
Yeah.
And we have the same thing in the Caribbean. There's these waves of ceramic technologies that come through, and people are like, oh, there's mass migrations from the south, and it's like wiping out these populations. And no, they're the same people. Geneticists come in, you look at them, they're the same people. They just, if you got a cool new fancy pot, why wouldn't you use that, right? Like make your other ones are breaking. It's time to move on. I like that I like that design. I like that vessel shape. They're gonna change.
So it's harder for us to imagine how something so drastic as giving up your monumental architecture, how can that happen? So we don't like to imagine anything internal, we like to imagine it external. That's not really what we're talking about here. I mean, that's not what we're here to discuss. I'm sorry. Taking you off on a lovely tangent.
Well, I think with both Manjusha and Sophie here, working with big scale kind of issues and so forth, I just wanted an issue, of course, that's particularly interesting to me because that's sort of the time period that I work in. But I'm just wondering, again, we have really small samples, and now that we're developing even more the ability to extract DNA from sediments, which is to me almost revolutionary, that we don't have to have human remains or other kinds of remains from animals or whatever. And that's really added a huge new dimension. But I just wondered where you too, with working with the big pattern, where you think we're going next. I mean, what's the next sort of big question? Is it collecting more samples primarily? And how are people feeling about the ability to accept changes in the interpretation? Because there are, I mean, ongoing, there are changes in, oh, wait a minute, or maybe, oh, the Denisovans are much more important now than they were a couple of years ago, right? In terms of admixture and what we have and so forth. I mean, in some recent account, it seems to me that, people are saying, oh, Denisovans were much better than Neanderthals. They've already sort of made a value judgment on which groups. Anyway, so what do you see in the big picture kind of things that might be where you might be going next?
I can, yeah. I have to say that ancient DNA has I don't know, many of you work with NGS data, we call it the NGS boom, which is like genomic data boom.
Right.
At some point, people spend billions on constructing human reference genome, and then now there's like every one of us can have genomes right now, sequence like this. Similarly, I saw the boom when as I said, when I started PhD, was just one high coverage. Now there's so many of them, and then the whole Holocene. Like I worked on a method we applied for dating it, make sure Neolithic movement across Europe and steppe spread across Europe. We developed methods, and it's so much data. I think there are samples, of course, give more granularity and coarser picture. We need tools to work with it. When we developed the method dates, before that there wasn't any method which can work with ancient DNA data because it's just so degraded. We are working with, just to give you a perspective, let's say we have 3 billion ATGCs in our genome. We can only work with 200,000 because just there's no data. And when we developed the method, everybody's like, it's not going to work. And then we did simulations to show it works. We made inferences, which aligns with archaeology. So I think the computational tools play an important role for interpretation and for understanding, mainly leveraging the data we have. Like we can have 100 samples and we need the tools, right tools. Like I don't know, most of you probably use admix tools, like things that you can compare genomes for. So the tools are already being developed by many labs, like Priya's lab and many labs also like in Harvard and Leipzig.
Right.
I think the tools are very important. The data is getting generated a lot with like the advances in ancient DNA technology. Now I know that people are also using in Bank, they have like robots to do sediment analysis. Like they can do 7,000 samples at a time. I was like, wow. I remember they were doing that when I was finishing my PhD. Matthias Meyer was like setting up the lab, and I was like, I'm gonna do 7,000 samples at a time. Like, yeah, it's good for sediments. That's great, right? Like now you can. One of my friends, Vivian, she did the pioneer work of the sediment analysis, and it's almost like what we say, she's like sieving the whole world for like DNA. Like that's what you can do. But when you don't have the methods for it, like the DNA is just not fit. Sediment data is extremely degraded. Like you think ancient DNA is degraded. Again, a perspective. Let's say human DNA is 300 beads long. Ancient DNA is 50 beads long. sediment DNA is 25 to 30 weeks long, or even lower. And sometimes we don't even know it's coming from the hominin. It can be coming from somewhere else. And you might be thinking that you're digging in this layer, maybe the age is this, but that DNA has broken down from that layer to this layer. So there's like so many complications. It's almost like a metagenomic analysis. So one needs very robust computational tools, which can help leverage the data and interpret the data. And I think once we get there, I think it's going to be so good. I think there are many labs working on it, and we are going to get there. Yeah. That's what I think.
You want to add?
Yeah, sure. Yeah, so sediment DNA is very hot right now in ancient DNA, and Manju talked a lot about it, so I won't talk too much more, but I would say that it's like 5 years behind where we are with skeletal ancient DNA in terms of the methods that we have because of the degradation and everything. So that's what everyone's doing right now, is to try to play catch up to what we can learn from extracting DNA from a bone where we know the species already, because it's a whole different ballgame when you don't have a reference genome for what you're expecting. And so you need very different computational tools. So what I'll talk about for my point is that it can seem, especially if you're working a lot in Europe, for example, that the ancient DNA sort of database for what we have in representation of time periods is very saturated, because there are literally thousands of ancient genomes from Europe. And part of that is because of Eurocentrism in science, and part of that is because Europe is a relatively cold and dry place, and so DNA preserves better. There's reasons for that. But in the rest of the world, which happens to be where all my work has happened, we actually don't have that many ancient genomes still. So I think there's a large gap that's being filled continuously with just more representation from different populations. And the other piece of that too is that there's poor representation outside of Europe, even from modern people in genetic studies as well. And there's a lot of computational tools that need modern human reference genomes from diverse populations, because when you get to very granular analyses, sometimes it's not enough to try to use a modern European to do certain things to my ancient South American genome, and I really need a closer representation or a match of the haplotypes that I'm seeing in my ancient individual. And so the representation gap, I think, is something that's also coming along for different areas of the world outside of Europe, which is really exciting to see, and it will obviously illuminate a lot more things. I also, back to the question at the beginning of outliers, I think geneticists or population geneticists at least often like outliers because they're inspirational to us about like research questions that we should be asking. So if I see an individual who doesn't fit the pattern I expect, then I might ask an archaeologist, well, why might this be? Is there something in the material culture that we're missing? So yeah, I think that certainly sediment DNA, but also more representation from the entire world. Because if there's humans, there's burials, and there should be ancient DNA. So
yeah, even in genetic analysis, outliers are very interesting.
Yeah,
we did a present day population, Indian population. Indian populations are amazing. I'm not saying because I'm Indian. Not believe me. The diversity of language, food, culture, and genetics is amazing. Like there's a whole mix, right? There's reason that they are excluded in research because it's very hard to interpret and analyze. But people are going there now. So we did a pilot study of Alzheimer's patients in India. We found this one Kerala guy from South, always grouping with Delhi, and we're like, maybe this is a missampling. Maybe there's a missampling. It's just that he said he's from Kerala because he's from Kerala. But I think like his generation moved one generation before. Mm-hmm. So it just tells us it's just one tiny dot, but like whenever I do a PCA, this dot is moving up all the time. And like, why is this South Indian guy in the map? Because when you do a genetic analysis, you can see a very nice climb of distribution of Indian population, which aligns with geography actually, because we have very distinct ancestry, like two major ancestry from north and south, and it's like very nice mixture. And we have in south, we have a lot of ancestral South Indian ancestry called ASI, and then ancestral North Indian groups that we label them. And so basically you can see like a nice climb. They don't clutter like Europeans or Italians. We follow a nice climb. Mm-hmm. And the South Indian there was always going up. And then it tells us a story like it's just not it's not a sampling error. It's just that the he has a history behind it. But it's just an example of a present day, right? You can go back and look at the document or records. But in ancient DNA, you cannot do that. That means that there was a story, like they were talking about. Probably he moved here for somewhere. Probably there's something happened there. So we don't know. We don't have a time machine, except the DNA, which acts as a time machine. So yeah, outliers are very interesting.
Yeah, just to add to this, what Sophie had to say here about expanding the understanding, because we are so overrepresented with Europe, which is in and of itself a political and cultural problem. And I was very impressed with Johannes when he was at this little gathering we had last week when we had 2 campus visitors. When asked, what would you do with $5 million, right? And he said he would use it to build local capacity so that many of the populations who today are not interested in having somebody from Europe come and tell them who their ancestors were, because they figured it out in a big deal lab. They actually, he would like to see people developing locally the skills, the techniques, and so forth, so they can tell their own stories and use it. And I thought that was a very important aspect of all this, because we certainly know there are many groups that are not interested in yielding their modern day DNA in order to serve a Eurocentric, science-oriented kind of mission or activity. So I just wanted to add that. I thought that was very good of him to have done that.
I love that, because that's what I've been sitting with. Okay, as someone who works in North America
Right,
now,
yeah.
with a lot of my populations, the only things that there's a whole political process, obviously, with dealing with human remains in North America. And in these destructive studies, we are only doing on our Caribbean samples, and that's just because those are the only places we are allowed to do that. But we often work with indigenous populations and with the tribes, because we have to, because we're decent human beings, and that's the right thing to do. And we also work with historic populations, because we do a lot of historic archaeology and marginal, like unmarked graveyards. I mean, they're finding those every day. Like, we get a call every day about, you got to come out here and tell us what we've done. And you're like, okay, we'll deal with it. But this idea that people are just excited to be a part of the process or to answer your questions is just not the case. And you are never going to convince someone that whatever intellectual pursuit you're on is important if they, like you said, either have figured it out for themselves or they've decided it's not important, and it's their own personal heritage, it's their own personal, literal biological makeup. And so there are just some things that we will never know, and we will never have reference collections for. It's something that I see a lot in my current work, that, like we were talking about in Europe, people have a very different connection to the physical remains of the past.
Yeah, I would like to jump in on this topic as well, because working at a museum which struggles with a lot of colonial history and is trying to hire the indigenous scholars and collection managers to handle those collections, it was one of the first, and in reflection of, we need more data points, we need more data, the future is gonna look great, and we're excited not only about ancient DNA based on human remains or non-human, but also now on eDNA data. This is maybe one part of the story, but the other part of the story is I'm drawing here from one of the earliest talks that I've heard at the museum, and it was one of the indigenous geneticists based in Arizona State University, who came to the museum, and she said, before we wanted to sample our ancestors, but now we take the soil below the ancestors. And it was such a catchy moment that I thought, oh wow. But it was really kind of her point was building the local capacity. What Johannes says, she is now a trained geneticist. She has built her own lab. They're trying to answer the questions that they're trying to answer. For example, not necessarily about kinship that maybe a lot of other people care about, but how to heal diabetes, because a lot of diabetes studies have been done only on white European population, and now they would like to do it really properly through their indigenous genetic data, and find new solutions to look into the future, and kind of also then build the data banks and protect them from open source publishing, which I think also doesn't go along with a lot of Western views of let's make all the data open access and share it and have this public knowledge. but kind of have this duality and allow it to happen, because these communities have been really underprivileged for so long, and we need to be able that sometimes we will not get all the data because local communities just won't want to collaborate or won't want to that data to be put into open source by others who can reuse it in many different ways. So that's just my take into ethics or
It's the reality.
Yeah.
Any other questions or comments? You have these wonderful, I mean, I'd love to hear each one of them give an hour or more talk. Maybe not right now.
I'll piggyback on that just real quick. Just as one thing we haven't said is that one of the onus on us as people who are getting this data and interpreting it is not only to honor the communities that we're taking this data from, but also the way that we present it to the public. And to be able to present it in a way that it can't be, as we were talking about, used for nefarious means by people who might want to take a more nationalistic stance about the connectedness between peoples and to the past and who can and can't do things. So I don't know if you guys, like, again, in my work, because I interconnect with the public so much, I don't know if I'm always nervous about putting those results out there about people who could be connected and making sure that I'm wording it in a way that is sensitive, but also can't be weaponized, I guess, even though I feel like it's just impossible. People can weaponize anything, but I don't know if you guys deal with that. I would like to hear from the other panelists or something, or people who might deal with that. How you handle it.
Yeah, we had a whole discussion about it, at least so that method, one of the method we developed dates is like, basically you can take a single genome and you can ask, when did the admixture occur in your ancestors? So you can basically use another tool and say, let's say I'm from South India and I know a little bit about my history. And then I can say, well, given, ancestral South Indian and ancestral North Indian, when did admixture happen in this Telugu individual? Just for 1 0 can do that. And it gives you a time frame, and it gives you an error, because you're not perfect. Like, it's not, because although I have like good genome, like it gives you still an error. The more individuals you have, more statistical power, you get more. This can be used for any purpose, right? For whatever you want. One of the ethical things that we were discussing at the point is that using it on populations which can be a target for whatever reasons can be harmful. And we didn't want to, like, there were some populations in our data set. I don't want to mention names, that can be politically incorrect. And at some point we decided, like, we are not going to talk about any of the present day populations. Of course, it's applicable for present day populations, but there are definitely, even today, right now, there are groups that are being segregated or discriminated based on whatever reasons, whatever ancestry they have, whatever ethnicity they have in different parts of the world. And there is no criteria for, bullying. Yeah. You just need something. And we never wanted our tool to be one. Again, like you said, like, people can, once you do something, there's always onus of us will be. So we always, of course, like, our whole paper was meant to show in ancient Indian, the power of the advantage of the method was it works beautifully on ancient DNA. So that's what we did. That's what we showed. And we showed that, for the first time we have a time of, when step pest is formed, we know that these populations were there, like, what is the ancestry, that Caucasus ancestry, okay, they formed there, and then this is the first time we have this time. So all that was good, but like, we made a conscious choice to, like, not show results. I wouldn't say it's like hiding results, but it's more like it was not useful, because we are highlighting the method, we are showing the power of the method, we are showing the usefulness of the method, and we didn't want to show in a present day population. So that's a choice we made. Maybe some people might not like it, but that was something I said, no, don't do it. Because you never know. Even if single person is harmed, it's not worth it. Right? Like, the idea of the method is to, like, make good progress in science, never to. At least that was experience I had.
Anybody want the final word? Yeah, maybe we'll say thank you.
Well, yeah. Say thank you, first of all,
to our panelists for sharing their work and sharing their insight into this. I also want to thank you all again for being here, but also celebrating this young generation of scholars who are going to be dictating our future. I'm so excited about that, and thinking about all the ways that this work will be applied and interpreted in positive and constructive ways going forward. So thank you all very much. Great job.